Good day, future doctors. So this is the first part of our lecture on the basic principles of serology. Let us start our lecture on defining some terms. So we have the terms analytical sensitivity and clinical sensitivity. A method is analytically sensitive if it can detect even the smallest amount of the analyte that you want to determine or measure in the sample of the patient. A method is analytically sensitive if it can diagnose the disease in its early or acute stage. And when you say that the disease in its early or acute stage, the amount of antigen and antibody are still very small. But if the method can detect them, then the method is said to be analytically sensitive. A method is clinically sensitive if it can produce a positive result if the patient really has the disease. So as much as possible, the method must not produce false negative results. Next are the terms analytical specificity and clinical specificity. A method is analytically specific if it will only detect the analyte that you want to determine or measure in the lab. So therefore, there is no presence of other interfering substances. So if you want to measure glucose in the blood of the patient, the other carbohydrates present in the sample of the patient must not react with the reagent of that method. A method is clinically specific if it will yield negative results if the patient does not have the disease. So as much as possible, there should be no false positive results. Next are the terms seroconversion and seroreversion. When you say seroconversion, this refers to the time it would take for the antibodies against a specific antigen or disease would become detectable in the sample of the patient. The usual sample that we are using in the laboratory is the serum. So the time it would take for the antibodies to be detected in the serum of the patient, that's seroconversion. Before achieving seroconversion, the serum or blood is labeled as seronegative. That would indicate that the serum does not yet have the detectable levels of antibodies. After seroconversion, the sample now will be labeled as seropositive. Seroreversion, on the other hand, is the opposite of seroconversion. This is the time when tests can no longer detect antibodies in the patient's sample. So if you have already received your COVID-19 vaccination, the seroconversion would be 2 weeks after the second dose. The seroreversion for some vaccines is 6 months after the second dose. So that would only mean that the antibodies are detectable 2 weeks after the second dose, and there's a chance that they will no longer be detected in the sample of the patient 6 months after the second dose. We will now differentiate qualitative from quantitative tests. When you say qualitative tests, these are tests that measure the presence or absence of the substance. With these tests, we are not concerned with the concentration of the antigen or antibody. We just simply want to know if the antibody or antigen is present or not. So the results are not expressed in numerical terms. Instead, we will use descriptive terms, such as what? Positive or negative, reactive or non-reactive, normal or abnormal. But if we want to know how much of this antigen or antibody is present in the sample of the patient, then we should do quantitative examination. The left photo is showing us the anti-streptolysin O testing. The presence of agglutination would indicate that the patient has antibody against streptolysin O and the patient could be having streptococcal infection. The agglutination result will only tell us that the patient has the antibodies, but it will not tell us how much of these antibodies are present. So therefore, anti-streptolysin O testing is an example of a qualitative test or examination. On the right photo, you have there a glucometer which can give us an exact numerical value of the glucose in the blood of the patient. So therefore, the use of glucometer is an example of a quantitative examination or test. What is now semi-quantitative test? This type of test is similar to qualitative test, meaning you are more concerned with whether the antigen or antibody is present in the sample of the patient. What makes it different from qualitative test is that somehow the result will give us an idea of an estimation of the amount of antigen and antibody in the sample of the patient. I will explain that one by giving you an example. A good example of a semi-quantitative test is the urine dipstick testing for the different analytes present in the urine. You have here a strip, and within the strip you have their pads. Each pad is designed to measure a particular analyte in the urine. I want you to focus on the protein. If that pad of the protein will be immersed in the urine, and if proteins are present, chemical reaction will occur or take place. If there are no proteins in the urine of the patient, then the pad will remain yellow as shown in the picture. If there are proteins in the urine of the patient, chemical reactions will occur, and that will cause the change in the color of the pad designed to measure protein. If the pad will turn from yellow to green, that will tell us that proteins are present, and that's basically qualitative testing. But the intensity of the green color will somehow give us an estimation of how much proteins are present in the urine of the patient. That will not give us the exact numerical value. That will only give us an estimation. So this test is a semi-quantitative test. It will tell us that the protein is present, and it will also give us an estimation how much of these proteins are present. It's very important for a student who is studying serology to know what are the bonds that would bind an antibody to an antigen. We're supposed to make them bind to each other so that we can detect them in the different methods that we will utilize in the laboratory. So what are these bonds? Covalent bonds are strong bonds, and they are usually formed with the sharing of electrons between atoms. So take a look at the picture of hydrogen and chlorine. The hydrogen will share its electron with chlorine, and vice versa. After the sharing of electrons, they will now be able to form what we call covalent bond. Unfortunately, covalent bonds are not involved in the interaction between antigens and antibodies. That would only tell us that the bonds involved in the binding of antigen to antibody are all weak types of bonds. The bonds that make antigens interact with antibodies are the following: hydrogen bond, electrostatic forces or electrostatic interaction, van der Waals forces, and hydrophobic bonds. Remember, all of these bonds are weak types of bonds. The first of these weak bonds is the hydrogen bond. Take a look at the picture. The blue arrow is pointing to an antigen. The yellow arrow is pointing to an antibody. And then take a look at the red arrow. The red arrow is pointing to the hydrogen bond. Now, how do we define a hydrogen bond? It's a bond that is formed between hydrogen and oxygen, nitrogen, or fluorine. As long as the bond would involve a hydrogen, you will call it as a hydrogen bond. So in this case, the hydrogen of the antibody formed a hydrogen bond with the oxygen of the antigen. The red arrow is pointing to the fragment of antigen binding portion of the antibody. As you can see, the Fab portion of the antibody is interacting with the green colored antigen. Take a look at the second red arrow. What made the antigen interact with the Fab portion of the antibody is the bond pointed by the red arrow, and it is formed between hydrogen and oxygen. So therefore, that is a hydrogen bond. The second type of bond involved in antigen-antibody interaction is the ionic or electrostatic interactions. This type of bonds arise from two groups. They are referring to antigen-antibody, and these two groups have opposite charges. So basically, if your antigen is positive and if your antibody is negative, then they will be attracted towards each other and they will form what we call electrostatic interaction. Please refer to the red arrow. As you can see, the fragment of antigen binding portion of the antibody has positive charges, while the green colored antigen has negative charges. With the opposite charges, they are now attracted to each other, and the bond that is formed is called electrostatic or ionic interaction. The third type of bond involved in the interaction between antigen and antibody is the hydrophobic band. Please take a look at the photo. You have there the pink-colored antigen and the blue-colored antibody. Now, hydrophobic bonds are formed every time that the antigen and the antibodies will interact to seal away the hydrophobic components from water. So what do I mean by that? So I want you to take a look at this first red arrow. That's the hydrophobic component of the antigen. And the antigen must look for a way to not expose this hydrophobic component to water, because when you say hydrophobic components, they do not interact well with water. This is the hydrophobic component of the antibody. To keep these hydrophobic components away from water, the antigen and the antibody will bind together so that these hydrophobic components will be sealed off away from water. So as you can see in this picture, hydrophobic bonds are formed between the hydrophobic components of the antigen and antibodies in order to keep them away from water. Hydrophobic interactions describe the relations between water and the hydrophobic components of the antigen and antibodies. The last but not the least is the van der Waals force. Take a look at the two blue arrows. They are pointing to the antigen and antibody. And what do you notice? There is the presence of cloud of electrons surrounding the antigen and antibodies. Van der Waals force happen if this cloud of electrons will shift on one side of the antigen and antibody. So take a look at this antigen. It's very obvious that on its right side, you have there the cloud of electrons, while it has the protons on its left side. But on the case of the antibody, you have there protons on its left side and electrons on its right side. So what will happen now? Since the left side of the antigen has electrons and the right side of the antibody has protons, these protons and electrons will be attracted to each other, and they will now create what we call van der Waals forces. So van der Waals forces result from transient shift in the clouds of electrons surrounding the antigens and antibodies. So let's try to identify the type of bond described in each of the statement. This is a bond that is formed between hydrogen and oxygen, or hydrogen and nitrogen. So this is the hydrogen bond. This bond is formed by the redistribution of clouds of electrons. Yes, this is van der Waals force. How about water-insoluble components are coated by polar components? You have here the clue already, no? Water insoluble components. So this one is the hydrophobic interaction or hydrophobic bond. Positively charged particle attracted to negatively charged particle. This is ionic or electrostatic interaction. The ratio between the antigen and antibody will have an impact on the interaction between these two. Should there be an excess of any of the antigen or antibody, then the result can become false negative. Now, that is best explained by these three zones. We have prozone, zone of equivalence, and post-zone. Zone of equivalence has the optimal ratio of antigen and antibody, thus expect to find a positive result. While in prozone, you have their excess antibodies. And what will happen? That will now produce a false negative result. In the post zone, it's very obvious in the picture, there is an excess of antigens over the antibodies. So for you to easily remember that prozone has excess antibodies, I want you to take a look at the picture. Every time we're doing that, we're actually expressing strength. And when you have excess antibodies, you are actually strong. I am pro. I am strong. Excess antibodies. In order to produce a positive result in a serological test, the ratio of the antigen to antibody must be within optimum range. That is to make it possible for the antibody to cross-link one antigen to the other to cause the formation of a lattice. In the case of Prozone phenomenon, there is excess antibody level. So these antibodies, Prozone phenomenon results to false negative result secondary to a higher antibody titer compared to the antigen. The high amount of antibody present in the sample will cause all of the binding sites in the antigen to be occupied and coated, thus making it impossible for the antibodies to cause lattice formation with the antigen. We have here a case of Prozone phenomenon. We have samples 1 and 2. Both samples were serially diluted. Expect that the test tube with the 1:512 dilution should contain the lowest amount of antibody because the sample there is very diluted. Now, take a look at sample number 1. If undiluted, it was able to produce a 4+ grading of agglutination. As soon as you dilute the sample, you will notice that the grading of the agglutination will also decrease. Let's check sample number 2. If the sample is undiluted, you won't appreciate any agglutination. That could only mean two things. Number 1, the patient does not have the antibody because the patient is not having the infection. Or number 2, there's a probability that this is a false negative result. If the sample is serially diluted, you will notice that the sample was able to produce a 4+ grading of agglutination if it was already diluted 1:4. That will only tell us that you need to dilute the sample first before you will achieve the optimum ratio between antigen and antibody. So meaning, when the sample was undiluted, the antibody titer is higher than that of the antigen. You will only appreciate the optimal ratio of the antigen and antibody, thus causing lattice formation in the sample that was already diluted. So sample number 2 is an example of a prozone phenomenon. So I want you to imagine that you're already working as a medtech and you are confronted by a doctor because the result that you release for anti-streptolysin O is negative, but the doctor is convinced through his medical history taking and physical exam that the patient is suffering from streptococcal pharyngitis. If you are suspecting that the false negative result is because of Prozone phenomenon, what should you do to address the problem? You know very well that in Prozone, there is excess antibody. So the best thing that you should do is to dilute the sample of the patient. If the diluted sample will produce a positive result, then you can explain to the doctor that the result that you initially released was a false negative result, and it's primarily because of Prozone phenomenon. In post-zone phenomenon, there is excess antigen, and the number of antibody is not enough to cross-link all of them, resulting to a false negative result. So post-zone phenomenon results to a false negative result because of the higher antigen titer compared to the antibody. So the amount of the antibody present in the sample is not enough to cross-link the antigens and produce lattice formation. So let's say you are again confronted by a doctor because of the negative result on ASO that you released to the patient. The doctor is convinced that the patient is a case of streptococcal pharyngitis. If you are suspecting that the result that you release is a false negative result due to post-zone phenomenon, what do you think you should do? So, can you please stop the video temporarily, and I want you to think of a way on how to address a post-zone phenomenon. Remember, in this phenomenon, there is excess antigen titer. Take note, in Prozone phenomenon, you can just simply dilute the patient sample. But in post-zone phenomenon, you cannot dilute the reagent because every time that you will do that, there's a probability that the reagent will be contaminated. So we do not dilute the reagent containing the antigen in the laboratory. So if you cannot do the dilution of the reagent to address the problem of post-zone phenomenon, how will you now correct a post-zone phenomenon or a false negative result due to post-zone phenomenon? Okay, please stop the video and think of a possible answer. Now, how to solve a post-zone phenomenon where there is excess antigen level? Remember, we need to get the optimum ratio of antigen and antibody. And what is lacking in post-zone phenomenon is the antibody level. So, we need to repeat the test days or weeks after the initial testing. That is to give time for the patient's immune system to produce higher amounts of antibody. And with the increase in the antibody level in the serum of the patient, we might achieve the optimum ratio of antigen and antibody titer. So let's have a review. To correct the problem of Prozone phenomenon, we just simply dilute the patient sample to lower the antibody titer. Because the problem in Prozone phenomenon, there is excess antibody. To correct the problem of post-zone phenomenon, we need to repeat the test days or weeks after the first testing. That is to give time for the patient to produce enough number of antibodies to achieve the optimum ratio for antigen and antibody. At this point, we will classify the different serological tests that we can perform in the laboratory as to primary, secondary, and tertiary. When you say primary serological tests, the reactions of the antigen and antibody in this type of test are not directly seen by the naked eye. Because there's no way for us to see antibodies and antigens because they are always soluble. Not unless the antigen is attached to a cell or a particle. But if not, both of them are invisible to the naked eye. So for us to see if the antigen is now bound to the antibody, we need to add labels. So primary serological tests would require the use of labels to make the reaction directly observable. What makes this primary serological test better than that of secondary is that they have higher sensitivity, meaning they can detect even the smallest amount of antigen or antibody present in the sample of the patient. These are the three primary serological tests. We have the enzyme-linked immunosorbent assay or ELISA. The label that is used in this test is enzyme. Take note of that. We have immunofluorescent antibody technique. And the label that is used here is a fluorescent chemical or fluorescent dye. And lastly, we have Regio Immunoassay. And the label that is used here is a radioisotope. All of these tests have higher sensitivity than secondary serological tests. But if you will be asked which among them has the highest sensitivity, the answer is Regio Immunoassay. Dengue Duo Test Kit is an example of a primary serological test. Without the formation of that pink color, there's no way for us to determine that there is that antigen and antibody interaction. Now, I want you to take a look at the test kit. Please look for the area labeled as S, that is the sample well. And then take a look at the areas labeled with the letters CTR, that's the control. Then we have IgM zone, and we have IgG zone. So I want you to recall the picture of the Dengue Duo Test Kit. You have here the blue circle representing the sample well. Then you have the three zones, the IgG zone, the IgM zone, and the C zone or the control zone. Take a look at the components of the test kit. In the G zone, you have there black-colored Y-shaped structures which are actually anti-IgG. So these are antibodies that will capture IgG. These anti-IgG are immobilized within the G zone. Immobilized in the M zone are anti-IgM. So these are antibodies that will capture IgM in the sample of the patient. In the control, you have their anti-dengue antibodies. So meaning these antibodies will capture dengue virus antigens. I want you to focus on the sample well. Do you know that present in the sample well is the recombinant virus? When you say recombinant virus, that particular virus will contain all the serotypes of all the antigens of the serotypes of dengue. And this recombinant virus is labeled with an enzyme. Again, in the sample well, you have their dried recombinant virus carrying now the antigens of all the four serotypes, and it is labeled with an enzyme. So let's say we have a patient who is in the early stage of the dengue infection. So expect that the patient has IgM in his or her serum sample. So what we will do is to add the serum of the patient into the sample well. So expect the red-colored Y-shaped IgMs of the patient will bind to the viral antigens present in the sample well. Take note, the manufacturer of this test kit will always make sure that the number of the antigen present in the sample well will always be higher than the antibody of the patient. And what will happen now? The antigen, the free antigen, and the bound antigen will diffuse towards the three zones, the G, the M, and the control. So there is capillary diffusion. The free and the bound antigens will just simply diffuse through the G zone because the G zone is supposed to capture IgG antibodies. The diffusion will continue until such time that the bound and the free will diffuse through the M zone. Remember, immobilized in the M zone are anti-IgM antibodies. And since the bound form of the viral antigens are coated with IgM, they will be trapped along with the IgM in the M zone. The free viral antigens will just simply diffuse through M and will continuously diffuse towards the control zone. And since immobilized in the control zones are anti-dengue antibodies, the free viral antigens will all be captured in the control. Now what will happen? The enzymes present in the viral antigens will produce the pink color. So since there are viral antigens trapped in the M zone and also in the control zone, expect to find pink lines on these zones. And this is now how the test kit will look like after the incubation period. You will have pink lines on both control and M zones. Without the incorporation of the enzyme, the reaction will not be visible to the naked eye. Thus, this is an example of a primary serological test. Let's see what will happen if the patient does not have the dengue infection. So, after adding the patient's sample into the sample well, the free viral antigens will diffuse towards the G zone. They will just pass through the G zone without any of them being captured. Then they will continuously diffuse towards the M zone and none of them will be captured. And all of these free viral antigens will be captured in what zone? Control zone. So expect that the pink line will only develop on the control zone. It's very important for all of the test kits with this type of setup to have pink lines on the control zone. If the pink line will not develop, you should repeat the test. So for the reactions in primary serological test to be visible to the naked eye and for the reaction to be measured, labels should be added. So a label is a molecule that will react as part of the assay to make the reaction visible, observable, and measurable. These are the examples of the labels. For Regio Immunoassay, we have radioisotopes or radioactive compounds. And in order for us to measure if there is reaction of the antibody and antigen, we'll measure the level of radiation. In the case of ELISA, we're using enzyme. And we expect that there is production of color. Then in the case of immunofluorescence, we will be using fluorescent dyes or fluorescent chemicals. Then we will detect light production or fluorescence. Listed here in this PowerPoint slide are the different radioactive compounds that we can utilize in Regio Immunoassay. So please memorize them. And for us to determine whether there's really antigen and antibody interaction in Regio Immunoassays, we need to measure the level of radiation. And we can only do that using gamma counters or scintillation counters. And as mentioned a while ago, that among the three primary serological tests, the one with the highest sensitivity is Regio Immunoassay. The problem with this method is that since the person who will be processing the sample will be exposed to radiation, so there is health hazard, you need sophisticated instruments and machines. You also need to have the person who will process the sample undergo training. And you also have problems with how to dispose the radioactive compounds. So these tests are very expensive and there is occupational hazard as well. Regio Immunoassay makes use of radioactive compounds as labels, and these radioactive compounds will emit radiation, and the emission of radiation would indicate that there is the reaction of antigen and antibody in the test. the amount of radiation will reflect the amount of antigen or antibody present in the sample of the patient. The amount of radiation is measured by scintillation counters and gamma counters. Shown in the picture is a scintillation counter. As you can see, the scintillation counter will detect the high energy photon or radiation coming from the Regio Immunoassay setup. This scintillation counter has this particular part that is labeled in the picture as scintillator. The high energy photon or radiation will hit the scintillator, and the main purpose of the scintillator is to convert the radiation into light energy. It does it by releasing electrons. As you can see in the picture, there is the escape or release of electrons from the scintillator. The problem with the scintillator is that the amount of light energy that it produces is not as much as the amount of radiation. Remember, the amount of radiation will reflect the concentration of the antigen and antibody. So the light that must be detected by the scintillation counter should match the energy of that of the radiation. So this instrument must look for a way or must come up with a way in order to match the amount of light energy to the amount of radiation that is coming from the Regio Immunoassay test setup. That's why, as you can see in the picture, once the electrons are released from the scintillator, they will come in contact with the photomultiplier. So look for the photomultiplier tube name in the photo and take a look at the portion of the instrument that is labeled such. So, inside the photomultiplier, you have there blue-colored diodes. One of them is pointed by the red arrow. What will happen is, once electrons are released from the scintillator, they will come in contact with the first diode. Diodes are very sensitive to electrons. When they are interacting with electrons, they will release more electrons. That's why, as you can see, one electron hit the first diode and 3 electrons will emerge from it. And the 3 electrons will come in contact with the second diode. And what will happen? From the second diode, you have there 5 or 6 electrons jumping towards the third diode. So the purpose of increasing the number of electrons is to increase the amount of light energy so that it will match the amount of radiation coming from the Regio Immunoassay setup. To make you imagine how the photomultiplier would work, just take a look at this red arrow. The red arrow is representing the electron that would come from the scintillator. So the electron will hit the first diode. And what will happen is electrons will jump from the first diode to the second diode. And the role of the second diode is to multiply the number of electrons. And what will happen as the electrons transfer from one diode to the other, they are multiplied. Thereby, the amount of light energy is increased. So scintillation is a flash of light produced in a transparent material that is called scintillator if that transparent material is hit by a high energy photon or radiation. A scintillation counter is an instrument for detecting and measuring ionizing radiation by the excitation effect of the incident radiation on the scintillator. And there is that resulting production of light energy. The label that is used in enzyme-linked immunosorbent assay is enzyme. And since there is production of color with the reaction of the enzyme on its substrate, the reaction is measured and read by spectrophotometry. The next question is, what enzyme or enzymes is or are used in ELISA? The two most commonly used are the following: horseradish peroxidase and alkaline phosphatase. Listed in the PPT are the other enzymes that are also used in ELISA, but I would require that you memorize the two most commonly used. Enzymes need specific substrates so that they can produce the expected color reaction in ELISA. In the case of alkaline phosphatase, the substrate is p-nitrophenyl phosphate. And from the name of the enzyme, this alkaline phosphatase will remove the phosphate from p-nitrophenyl phosphate, converting the chemical into p-nitrophenol or para-nitrophenol. Para-nitrophenol is a yellow-colored compound. So the N color for ELISA using alkaline phosphatase is yellow. Horseradish peroxidase makes use of tetramethylbenzidine as its substrate, and the N color is blue. Take a look at the chemical reaction behind the ELISA method using alkaline phosphatase. The substrate is para-nitrophenyl phosphate, which is a colorless compound. In the presence of alkaline phosphatase, phosphate is liberated with the subsequent production of para-nitrophenol, which is a yellow-colored compound. This is the chemical reaction behind the horseradish peroxidase. The substrate is tetramethylbenzidine blue. It is originally in its reduced form, and if it is in reduced form, it is colorless. In the presence of the horseradish peroxidase, the tetramethylbenzidine blue is oxidized to form the blue-colored compound. In the ELISA using horseradish peroxidase, the substrate is tetramethylbenzidine and the final color is blue. Using the spectrophotometer, one should set the wavelength at 620 nanometer because that is the wavelength that blue light will absorb another type of light. Since in most laboratories, they can use alkaline phosphatase and horseradish peroxidase as enzyme labels for ELISA, there's a need for the medical technologies to always recalibrate the wavelength set in the spectrophotometer depending on what enzyme is used. Remember, the final color for horseradish peroxidase is blue, while the final color for alkaline phosphatase ELISA method is yellow. So if you're using tetramethylbenzidine blue, you set the spectrophotometer at a certain wavelength. Then if you'll shift to alkaline phosphatase, then you also have to reset the spectrophotometer. Now, to avoid that problem, because there's a probability that there is misreading of the specimen, scientists have came up with a way on how to make the final color of horseradish peroxidase ELISA method become yellow so that there's no need for you to always recalibrate the wavelength of the spectrophotometer. You just simply need to add sulfuric acid and the blue color will become yellow. You have learned from Clinical Chemistry 1, particularly on the chapter of automation, that colors can absorb other types of colors. In the case of a red-colored suspension, once it is presented with different types of light, it will actually absorb green light. The darker the red color, the higher the amount of green light that it will absorb. So that's why it's very important for you to always remember that if you are performing spectrophotometry, you should have an idea of the final color of the product or the test because the type of light it will absorb will matter. So, if you're processing a red-colored solution or final product, it's important to set the spectrophotometer within the wavelength of the green light because that is the type of light or color that is absorbed by the red color. So these are the different visible colors. You have here the Roy G. Biv, red, orange, yellow, green, blue, indigo, and violet. And you have on the second column their specific wavelengths. Remember, a red-colored suspension will absorb green light. And according to this table, the wavelength for green light is 495 to 570. That's the very reason why if you're performing hemoglobin concentration determination in hematology, to set the spectrophotometer at 540 nanometer. Why? Because the wavelength for green light is 495 to 570. And that will include the 540 nanometer. When you are performing hemoglobin concentration determination, the final color is red because that's the color of hemoglobin. So expect that hemoglobin will absorb green light. So set the spectrophotometer within the green region. Let's review. The final color for the horseradish peroxidase method of ELISA is blue. And as mentioned a while ago, the spectrophotometer should be set at 620 nanometer. Why is it so? The complementary color for blue is orange. So blue will absorb orange, and as well as orange will absorb blue. So for you to determine the concentration of the blue-colored compound in the horseradish peroxidase method of ELISA, you set the spectrophotometer at the wavelength of the orange color. And what's the wavelength of the orange color? 590 to 620 nanometer. This kit is utilizing ELISA as its principle. But you've learned a while ago that the final color for horseradish peroxidase is blue, for alkaline phosphatase is yellow. So what is producing this pink color? The answer is colloidal gold conjugate. Unlike with other substrates used in ELISA, this one does not require an enzyme. So I just don't know why scientists classify this one under ELISA because this one doesn't need an enzyme. The point is, this is actually a gold conjugate, and once they are concentrated on one area, they will produce a characteristic red color. That's the very reason why the control zone and the other zones in the test kit would produce a pink to red color. It's because of the colloidal gold conjugate. In terms of immunofluorescence, we are actually utilizing two important dyes. One can produce red color, red rhodamine, and the other one can produce green color, and that's fluorescein isothiocyanate. Red rhodamine, green fluorescein, isothiocyanate. So it is the fluorescein isothiocyanate that was used in FDA ABS treponemal test for syphilis. Primary serological tests or primary immunoassays can be classified as competitive and non-competitive. When you say competitive, a known antigen or antibody will compete with the patient's antigen or antibody. So for you to understand that, I want you to take a look at this test kit. Immobilized in the test kit are the antibodies directed to the antigens that you want to measure in the patient sample. You can see in the picture that the patient does not have hepatitis B. So expect that the patient will not have hepatitis B surface antigen in his or her sample. Also added into the setup is a known hepatitis B surface antigen, right side of the photo, that is labeled with iodine, which is a radioactive compound. The sample of the patient and the known hepatitis B surface antigen with radioactive compound are added at the same time into the setup. But since the patient does not have the antigens for hepatitis B, the known hepatitis B with iodine radioactive compound are all free to bind to the antibodies. That's why, even if you will do washing, they will not be removed from the setup. And once you will measure the amount of radiation, you'll get a very high result. And that would indicate that the patient does not have the antigen. What if the patient has hepatitis B as indicated in the photo? So if the sample of the patient and the reagent with the known hepatitis B with iodine as radioactive compound are added at the same time, there will be a competition. Not all of the binding sites will be occupied by the known hepatitis B antigen. So after washing, some of these known hepatitis B surface antigen will be washed off. And what will happen? Once you will try to determine the radioactivity or radiation, the gamma counter and scintillation counter will measure or detect a decrease in the amount of radiation, indicating that something competed with the known hepatitis B surface antigen, and that would only mean that the patient has hepatitis B infection. So what I want you to understand, the higher the number of antigen or antibody in the sample of the patient, the amount of radiation, the amount of enzymatic activity, and the amount of fluorescence will decrease. Again, the higher the number of antigen or antibody in the patient sample, the amount of label that will be detected will be lower. The labels can be detected through radiation in Regio Immunoassay, color production in ELISA, and fluorescence in immunofluorescence. The lower the number of antigens or antibody in the patient sample, the amount of label that will be detected in competitive primary serological tests will be higher. Primary immunoassay can also be non-competitive. So there's no need for a known antigen or antibody to compete with the patient's antigen or antibody. And non-competitive primary serological tests can be classified as indirect, sandwich, or direct. When you say non-competitive sandwich type, these tests are designed to detect the presence of antigen in the sample of the patient. So if you want to determine the presence of antigen in the sample of the patient, your setup should have immobilized antibodies. For you to understand non-competitive sandwich type, I want you to take a look at these two wells. The first well contains the positive sample. The second well contains the negative sample. As you can see in the picture, there is now the introduction of the patient sample into the well. Immobilized in the wells are antibodies. Please focus on well number 1. The patient sample in well number 1 contains the target antigen. Thus, you can see that the antigens are now bound to the immobilized antibodies. While on the second well, the patient does not have the antigen specific to the antibodies. That's why the other antigens will just remain suspended in the solution. The next thing that should be done is to aspirate back the excess antigens. This is done by performing washing. Remember, the antigen and antibody present in the wells are not visible to the naked eye. That's why we need to add or incorporate labels. In this case, we will add antibodies labeled with enzymes. And we will make it a point that the antibodies are specific to the antigen that we want to test in the laboratory. So as you can see in well number 1, the newly introduced antibodies with the enzyme are now bound to the antigen of the patient's sample. While the newly added antibodies with the enzyme label in the second well remain suspended in the solution. The next thing that we will do is to do washing. So expect that the antibodies with the enzyme in well number 1 will not be removed during the washing procedure because they are attached to the patient's antigen, while all of the antibodies with the enzyme label in the second well are all washed off. Then we will add the substrate. So expect that the substrate will be acted upon by the enzymes in well number 1, and that will now produce a colored compound, indicating that the patient sample has the target antigen. While since you do not have the enzymes in well number 2 because the antibodies with the enzymes were all washed off, expect that the substrate will not change in color. So as you can see, there is no competition in the principle of this test. That's why this one is called non-competitive primary serological test. And take a look at the antigen. It is sandwiched between two antibodies. That's why this is non-competitive method sandwich type. Aside from sandwich type, we also have indirect type of non-competitive immunoassay. This time, it is designed to detect the presence of antibodies in the patient sample. So what should be immobilized in the sample wells? The antigens. Just like in our previous discussion, well number 1 has the positive sample, well number 2 has the negative sample. So expect that the patient's antibodies will bind to the specific antigens immobilized in the sample well. This will be followed by washing. This is the completion of the washing. And since the interaction of the antigen and the antibody is not yet visible to the naked eye, we need to add a second antibody. And this antibody will attach to the patient's antibody. And they bear with them an enzyme. These antibodies can also bear fluorescent dye or radioactive compounds. That will now depend on the method that you want. So this one is non-competitive indirect ELISA because the label used is an enzyme. So the antibodies with the enzyme label will all bind to the patient's antibodies. Washing is done after that. Expect that only well number 1 will contain the enzymes because the antibodies with the enzymes are bound to the patient's antibodies. So it is only in well number 1 that there would be what? Color production upon the addition of the substrate. Let's review. Sandwich type is used to detect antigen. Indirect type is used to detect antibody. The last but not the least, the non-competitive direct type. It is named direct because you will just simply add the antibody carrying the label into your sample, and the main role of that antibody is to look for that specific antigen. So for you to appreciate that, I want you to imagine that a tumor was removed from the body. If the pathologist would want to know where are the tumor cells in the cross-section of that tumor, the section of the tumor will be added with antibody carrying an enzyme, a radioisotope, or a fluorescent dye. And what will happen? So let's say the pathologist used antibody with fluorescent dye. So after adding the antibody with the fluorescent dye into the cross-section of the tumor, the pathologist will view the specimen under fluorescence microscope. And what will happen? If the tumor cells are now bound to the antibodies with the fluorescent dye, those tumor cells would produce fluorescence under fluorescence microscope. So you see, this method does not include washing because all you have to do is just simply add the antibody with the label, and the role of the antibody is to look for the specific antigen. If the method does not require washing, you refer to it as homogeneous immunoassay or homogeneous serological test. But if the method requires washing, that's the very reason why most of them are time-consuming. They are referred to as heterogeneous immunoassays. Since this method did not require washing, so this one is a homogeneous immunoassay. while this one involves washing to remove unbound antigen or antibodies. So therefore, this is an example of a heterogeneous immunoassay. The last topic, the difference between Regio Immunosorbent test and Radioallergous sorbent test. Now take a look at this setup. You have here immobilized anti-human IgE. So these antibodies are designed to capture the IgEs in our serum. So, after the addition of the serum of the patient into the setup, all of the IgEs will be captured. And since the interaction between the antibodies are not yet visible to the naked eye, we will add a third antibody. And this third antibody is labeled with an radioactive compound or a radioisotope. Then, we will measure now the amount of radiation. The amount of radiation will reflect the amount of IgEs in the patient sample. Now take note, Regio Immunosorbent test is designed to measure the total IgE level of the patient, and that can suggest possibility of allergic diseases or conditions. But the thing is, radioimmunosorbent tests will not determine to what the patient is allergic to. It can only tell the doctor that the patient has allergic conditions. On the other hand, what is immobilized in the setup of radioallergous sorbent tests is a specific allergen. So expect that after adding the serum of the patient into the setup, only the IgEs specific to that allergen will be captured. So, with the addition of the third antibody carrying the label, the amount of radiation will only reflect the IgE of the patient directed to that specific allergen. So, radioallergous sorbent test can determine the level of specific IgE antibodies, while radioimmunosorbent test is a technique that is used to measure the total IgE of the patient. Again, radioimmunosorbent test measures total serum IgE, while radioallergous sorbent test measures the level of specific IgE antibodies. And that ends my lecture on Introduction to Serology Part 1. Thank you very much for listening. Sites in the antigen, making it impossible for them to cross-link one antigen to another.